Hello friends, good morning. Welcome to Sam Biology class. Today we shall discuss about the principles, procedures and the applications of polyacrylamide gel electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Polyacrylamide gel electrophoresis, known as PETS, is a technique widely used for the separation of charged macromolecules such as proteins in a matrix of polyacrylamide. It also offers high degree resolution for DNA fragments. Macromolecules are separated on the basis of their electrophoretic mobility, which is a function of their size, shape and net charge. In this technique, separation is carried out in a cross-linked polyacrylamide matrix. The gel matrix is a porous mechanical support that is used as a stabilizing medium. Because of the gel matrix, the convection current as well as the diffusion effect is minimized. This results in sharper resolution of sample components. The gel matrix is porous and hence it serves as a molecular sieve that contributes to the separation of biomolecules on the basis of their size. Polyacrylamide is formed from the polymerization of acrylamide monomers in the presence of a smaller amount of N-prime methylene bisacrylamide. Bisacrylamide, which consists of two molecules of acrylamide joined by a methylene group, is used as a cross-linking agent. Acrylamide monomers are polymerized in a head-to-tail fashion into long chains. Occasionally, a bisacrylamide molecule is introduced into the growing chain, thereby introducing a second site for chain extension. Polymerization in this way results in the formation of a cross-linked porous matrix with a fairly well-defined structure. To prepare polyacrylamide gel, ammonium persulfate and N', tetramethylene diamine TEMED are added to a mixture containing acrylamide and bisacrylamide Low pH and exposure to oxygen are prevented as it will inhibit the process of gel polymerization. The pore size can be controlled through the concentration of monomers and the proportions of the cross-linkers used. The concentration of acrylamide generally set in the range of 4% to 20%. Polyacrylamide gel electrophoresis is most commonly performed in a vertical mode that utilizes a discontinuous buffer system. The electrophoresis unit comprises two buffer chambers. The upper chamber has the provision of a cathode and the lower chamber containing the anode. The gel is polymerized between two glass plates and is mounted in such a way that the top of the gel is immersed in the upper buffer chamber and the bottom in the lower. Before polymerization, a plastic comb is placed at the top of the gel solution in order to create wells for sample loading. This is a vertical slab gel and these are the wells where samples are loaded. The sample is first mixed with a buffer which contains a tracking dye such as bromophenol blue and a dense compound either sucrose or glycerol. The tracking dye, which moves at a fastest velocity, facilitates the determination of the extent of electrophoresis. On the other hand, the dense compound ensures that the sample components are settled at the bottom of the well. This minimizes sample diffusion in the buffer solution placed in the upper chamber. The pH of the buffer solution is adjusted so that all sample components will have a net negative charge. Because of this, during electrophoresis, all of them will migrate from cathode to the anode through the gel. As already mentioned, gels are porous. The pores are formed due to cross-linking. During electrophoresis, gels serve as a size-selective sieve during separation. As proteins migrate through the gel in response to the electric field, smaller proteins will pass rapidly through the gel pores than the largest proteins. Because of this, smaller molecules will move at the fastest velocity through the gel, whereas the largest molecule will move very slowly. Due to differences in size, running through the gel for some time will result in the separation of charged sample molecules into distinct bands. These protein bands can then be visualized by staining with a suitable reagent such as Comasi Brilliant Blue dye. Polyacrylamide gel electrophoresis is normally performed in two different modes. First one is sodium dodecyl sulfate polyacrylamide gel electrophoresis, and the second one is native polyacrylamide gel electrophoresis. We will have a separate discussion on SDS phase and native phase later on. Now, applications of polyacrylamide gel electrophoresis. PES is used in many fields including genetics, molecular biology, biochemistry and biotechnology. It has several applications. Number 1. Forensic chemistry. PES is used for DNA profiling and identification and also in paternity test. Number 2. It is also used to purify and to characterize proteins and also to identify protein isoforms and variants. In addition to this, protein-protein interactions can also be studied with the help of polyacrylamide gel electrophoresis. One of the most important applications is the determination of molecular weight of proteins. Finally, it is also used to detect and quantify protein contaminants in foods and pharmaceuticals. Friends, this is all about polyacrylamide gel electrophoresis. For more updates, subscribe my YouTube channel, Sam Biology Class. Goodbye, good night.